Welcome, everybody, to FDN's Lab and Learn Show with Reed Davis, the founder and chairman of Functional Diagnostic Nutrition, and our science officer and chief of everything lab work, Dr. Aaron Gonshor, who is kind enough to spend some time with us today to go over one of my favorite labs. I've been running a version of this test on every client for over 25 years. We call it the Metabolic Wellness Profile or a panel. And it's got three really revealing markers about people. I'm not going to spend a lot of time because I just want to get to the nitty gritty and turn it over to Dr. Aaron. Sir, how are you today?
I am fine, and spring has finally arrived to the Northeast. There we go. So what I'm going to do to not lose any time is I'm going to go to this stuff right here. There we go. And do you see it? Everybody sees it?
Yes, sir. Looking good.
Very good. So as we've done in previous sessions, this is Lab and Learn. What we're doing is trying to familiarize everyone with the test, those who have never done it to actually see what it is, and for those who have done it over the years, just to give them some basics again in case there's some things that they have forgotten about, etc. And the way we've tried to structure this is just to do an overview. Clearly, each one of the analytes we're going to be looking at, each one of those can take 1 or 2 hours to discuss in detail. That is not the idea here. It's just to give you an overview. And this is going to be a case report that we're going to discuss later on. And this is what we do each time, is we take a case report in order to be able to see how the test would function. So we're going to discuss this case report a little bit later on.
Fantastic.
And so here we are. This is the Metabolic Wellness Profile. And as you said, Reed, it is very important. And it's interesting, you've been doing it for well on 25 years. A very important test. And the reason it's important is that it looks at 3 extremely important parts of our daily function: digestion, liver detox, and oxidative stress. And so we're going to go into each one of those in detail. This is what a typical report would look like with the first page on the left, and we're going to go through all of that. And like all of our reports, it then has many pages which go into the specifics and interpretation and commentary on each one of those analytes. And so let's take a look at digestion first. And what we're looking at is something called indican. Now, for those of a certain age who've been doing this stuff for many years, they'll say, oh, indican. We know about that. We used to do indican, and you would look for a change in the color of a liquid to see whether there was there was an indican issue. What is indican? Well, if you take a look at the intestine, if you get food intake and there's partially digested food, that food contains tryptophan. And tryptophan is not only in food, it's used by a lot of bacteria. That's the food they love to eat, is tryptophan. And at the end of the day, the anaerobic bacteria especially will break down tryptophan to create something called indole. And indole then gets taken through the portal circulation, which brings it over to the liver. And in the liver, it gets hydroxylated to create indoxal. Indoxal then gets conjugated with sulfuric acid, which then makes it indoxal sulfate. And then finally, it gets conjugated with potassium. And that's what indican is. Indican is actually indoxal potassium sulfate. And indican will then eventually go into the circulation, get taken by the kidney, and ends up in the urine as a measure of urinary indican. So bottom line, what is all of this? Well, what you really want to determine here is gut function, specifically dysfunction, dysbiosis, and if you have high anaerobic intestinal bacterial uh, presence, because that's what breaks down tryptophan to indole, etc., then you will know whether proteins are being sufficiently broken down. And if they're not being broken down properly, you end up with what we call putrefaction. And it's almost like what it means. It's not just putrid. You are not getting a proper breakdown of the proteins to the amino acid stage, etc. And so it is an important element in understanding whether or not the person is dealing with protein metabolism in the right way. And so indican and the conditions which create elevated indican, especially in the urine, the most important thing is dysbiosis. That's going to elevate urinary indican. The second thing that will do it is if you're taking in excess protein. If, for example, you're eating a lot of lectins, there are people who go crazy eating lentils and peanuts and on top of the meat and the wheat, etc. At some point, that's going to begin to have an effect. The body cannot break down all of that protein the way it should. The second thing would be everything is okay as far as protein intake, but you're just not digesting it properly. Why not? Well, it could be because of low hydrochloric acid, low HCl. That's hypochloridria, very well known. Or very low digestive enzyme production. A lot of people have problems in not producing enough proteases or peptidases, etc. So they're going to have that problem. You can have protein malabsorption. In other words, everything else is working well, but you're just not absorbing all of this properly. And an increased intestinal permeability, which in and of itself can be due to everything from nutritional deficiencies and toxins, let alone parasitic fungal infections, bacterial infections, etc. And then there's a plethora of different things which can cause it. And you need to understand that all of these, either individually or together, can cause major problems for protein metabolism, and that will show up as indican. Well, what gives you elevated indican? Well, one of the most important things that happens is the effect of dysbiosis, as I said, and that's SIBO. That's the overgrowth of bacteria in the small intestine. Very important, and I know many of you actually test for it. It is an important test. People with inflammatory bowel disease will have that. Those who have pancreatic insufficiency. Why is that important? Because they're not producing the enzymes properly, which are needed as part of the downstream metabolic effect on protein. I mentioned hypochloridria, insufficient HCl, people with ulcers, gastric ulcers, bacterial infection like H. pylori. You've all heard of that. Many of you probably do the H. pylori test, etc. So just remember, elevated indican levels in the urine is a byproduct of intestinal protein putrefaction. I'm not going to go into the great details. And if there is a urinary result which shows that it's elevated, well, what do? Well, one of the things you want to do, and on the left we talk about that, you want to optimize the level of stomach acids. And there are many things that you can do. We're not going into treatment here, but there are many things that you can do. You can replenish the digestive enzymes, and there is a whole series of things. And by the way, on our website, we have a manual that goes through all of those things. There are, of course, the supplements, the probiotic and the prebiotic supplements, even more important than the probiotic for gut support. And as I said, further testing, right? SIBO. You can do the comprehensive digestive stool analysis, CDSA, the mucosal barrier test, because you want to know, is there an inflammatory condition? Is there intestinal permeability? And what kind of effect is that having? And what are the possible investigations that you want to do? And this is very important because a lot of people that you're going to see are not going to tell you that they're on various types of over-the-counter medication. And two of them that are specifically important for you to know about are H2 blockers, one of the most famous proprietary names is Zantac. It reduces stomach acids that's produced by blocking the histamine receptors. And then the proton pump inhibitors, which are even more ubiquitous and actually quite effective for the people who have these problems. One of the famous proprietary names for that is Prevacid. And what does that do? It slows down the enzyme that's affecting stomach acid secretion. And so it's used to treat acid reflux, GERD, ulcers, etc. A lot of people are swallowing antacids like there's no tomorrow, but they don't tell you about it. They don't think it's important for you to know. So you need to ask those questions. And of course, there's no question if they have an intestinal obstruction, that's something that you want to know. So that was a quick view from 30,000 feet of this incredibly important aspect, which is digestion or maldigestion. What about liver detox? Well, remember, there are daily life overloads, as we say, which can cause liver dysfunction. There are some of the usual suspects: too much alcohol intake and smoking. I don't have to tell you that both of those, if you are going to do it, need to be done in moderation, to say the least. The prescription, I mentioned this before, over-the-counter medicines of all kinds, various infections or illness, acute or chronic. What if there's poor nutrition? What if the eating habits and the dietary habits are such that they are an important aspect of this? Various other things, and I've put stress down here, both psychological and physical. Do not discount that. That is extremely important, that we can give a whole lecture on the effect of stress on both digestion and liver function. Well, we talk about the liver as being the center of detoxification. But what's a toxin? What is it? Well, we know that it's a substance that disrupts proper body functioning. Well, how does it do that? Well, it can do that because there's maybe a toxin overload. You've taken something in which has just overloaded the ability of the body to deal with it. Or it could be something completely different. It's an undernourished body. The body, because of the undernourishment, can't deal even with things which would normally not be toxic, but become toxic because of the undernourishment. And of course, they may be bringing in food properly, but there's a compromised digestion. All of that ends up leading to less effective detoxification. So detoxification is nutrient dependent. It's very important, because if you're not taking in enough nutrients or not the right mix, you're going to have a problem with detoxification. So what is detox? Well, it's a biochemical process, and we're going to go through that in some detail in the next few minutes. You're taking non-water soluble compounds, and most compounds which come into the body are not directly at that point water soluble. In fact, many of them are just the opposite. They're fat soluble. They're not soluble at all. And you're trying to transform that into water soluble entities, which the body can deal with to really detoxify, and then to excrete them from the body in various forms. Yeah, urine, sweat, stool. So the benefit of detox is to protect the body from adverse effects of both external and internal toxins. There are exogenous and endogenous toxins. And this is a very busy slide, which I'm going to bring down, but this is what the liver does in its detoxification. It goes through what we call phase 1 and phase 2, and we're also utilizing the term phase 3. And what are we doing? We're taking toxins and we're bringing it over to what they need to be to become excretory. Well, what's phase 1? Phase 1 is really breaking down the toxin to begin with, because you've got exotoxins, you've got endotoxins, including a very important one, LPS, which we're going to have a separate session on. It's very important. And you take toxins which are actually lipid soluble. Lipid soluble means that they go into lipids very easily, but not into water. And you need to take those toxins And you need to use everything from enzyme cofactors to various reactions, oxidation, reduction, hydration, you name it, to bring it to an intermediate stage in which the metabolites of that are now more water soluble. They're not totally water soluble yet, but they're more water soluble. And what do you want to do? You want to get them to a point where these metabolites, these intermediates, can then be joined up. They can conjunctify, come together with everything from glycine and glutamine and taurine, all kinds of other substances that is going to allow you to then take that intermediate metabolite, which is still a problem, and turn it into a total water soluble excretory waste product. That's what the liver wants to do. And phase 3, of course, is the decision on which way to go. Do you go into blood and through the kidney to urine, or does it go into the gallbladder with the bile into the intestine and then come out finally as feces? Whatever. So bile acids is what is helping with all of that. And the levels in feces and blood and urine and biles, these are allowance as markers for various diseases, many different diseases, everything from hyperlipidemia to cholestasis, which is bile blockage, gallstones mostly, colon cancer, etc. And what we're going to do is show you this slide, another busy slide. Why? Because what we're trying to tell you is that bile acids are synthesized in the liver. And there's a total of 12 of them. And by the way, that's what we're giving you in the test, the 12 bile acids. It starts with cholesterol. And the cholesterol basically goes through a classic or alternate pathway. And what does it do? It forms two acids, cholic and chenodeoxycholic acid. So those are two. Then they get dehydroxylated to deoxycholic and lithocholic. So now you got four. Then they get conjuncted with glycine and taurine. And so you get all of these different combinations and you get another 8. 8 and 4 gives you 12. So you've got 12 bile acids that are in the kidney, in the liver, coursing through the body, doing what they need to do. And so you need to know, what are elevated total bile acids, TBA levels? What are some of the causes and what are the effects of that? Well, the causes are quite well known. If you have obstructive jaundice, cholestasis, acute or chronic hepatitis, liver sclerosis, and cirrhosis for people with high alcohol levels, or in the tragic cases of liver cancer, you can have that. Or what you see more often than you would think is something called BAM. And what's BAM? BAM is bile acid malabsorption. You're just not absorbing those bile acids properly. And these people get a very watery diarrhea, going to the bathroom frequently. It becomes pretty obvious. And the signs and symptoms of that are, again, well known: unstable glucose levels in the blood. They have problem losing weight. Whatever they do, they can't seem to lose weight. And they complain of poor metabolism. What do they have? Elevated LDL. Those are the low density lipoproteins. That's the bad type of cholesterol metabolite. Elevated triglycerides, reduced high density lipoproteins, which are the good ones. Indigestion, alcohol or fat intolerance. These people complain of headaches. They're irritable, depression, hormone imbalance. And many of them are going to give off quite a putrid body odor when things really get bad. And so you need to correlate that with liver cell damage due to what? Due to infection, due to various inflammatory conditions, etc. I'm not going to spend a lot of time on that. If you have low TBA, what could that be due to? Well, cholestasis. Cholestasis, meaning that you're not able to really produce all the bile acids that you need to. And so you get decreased secondary bile acids, those 8 that I was showing you at the bottom of that slide. Things like gallstones, cholecystitis, abscesses, all kinds of problems with the intestinal to hepatic circulation. The removal of a gallbladder. Some people never recover their full function and ability to be able to use the acids properly. And people who are obese, they have frank diabetes, possible cirrhosis, and malabsorption syndromes, they are going to have it. And the signs and the symptoms, you see them very, very often. People with inflammatory bowel disease that have these problems, diarrhea, very bad smelling gas. They constantly complain of stomach cramps and bloating, itchy skin. That's one of the first signs that you're going to have. And of course, the pale colored stool, quite often smelly, but pale colored stool. That is a classic sign for low TBA. And of course, you want to correlate that to digestive symptoms, etc. And I want to spend the last bit of time that we have on the oxidative stress. Now, this is arguably the most important part of this whole test. And it deals with DNA damage. And DNA damage is caused constantly, every second of the day. Everything from metabolism, inflammatory issues, the pollution around us, the pollution we cause ourselves in our body by smoking, radiation in its various forms, which can cause that problem, ultraviolet light, etc. And so a major cause of cell and DNA damage is oxidative stress. Now, what is it? Well, oxidative stress is the following. A cell every second of the day, in trying to produce oxygen to be able to have the mitochondria function, produces free radicals. And we're not going to go into what all of that is about. That's a separate lecture. It is produced in the cells during normal metabolic processes. There's nothing abnormal here. That's what happens. So the cell is always producing antioxidants to neutralize these free radicals, which is the byproduct of breathing, of the cell breathing, of using oxygen. So the imbalance of these, what we call reactive oxygen and nitrogen species, and the system's ability, the cell's ability to detoxify these reactive intermediates, is oxidative stress. And oxidative stress caused by these oxidative species is going to do a number of things. It's going to cause membrane damage to the cell. It's going to cause damage to the protein metabolism in the cell. And it's also going to cause problems to the DNA in the nucleus. And that oxidative damage can cause necrosis, apoptosis of the cell, pyroptosis, all the things which you don't want the cell to have to go through. So remember that if someone has chronic inflammation, and there are many discussions we can have about chronic inflammation, it is something we need to keep in mind at all times, is very much related to these reactive species. If you've got infections, if you've got tissue damage for any reason, if you've got inflammatory diseases, you've got colitis, you've got pancreatitis, you've got intestinal inflammatory diseases, you've got environmental oxidants like cigarette smoke and many other things, that's going to cause activation of inflammatory cells, and that in turn is going to end up causing free radicals to form in greater numbers than normal. And that's going to cause tissue and DNA damage. And it's going to basically alter the cell's functioning and its ability to grow. And so, as I said, there are a number of different types of reasons you get oxidative stress. One is endogenous, another one is exogenous, coming from outside, and one is the antioxidant dysfunction. It's just not creating antioxidant function well enough. And I mentioned before that you can have lipid issues, you can have protein issues, and DNA. And the markers for that can be markers of DNA oxidation or of protein and lipid. And we have chosen DNA damage, and the one element that we talk about here, and the biomarker that has been around for a number of years, that has become extremely well known as a very reliable biomarker, is 8-hydroxy 2-deoxyguanosine, lovingly called 8-OHDG. Now, it is a biomarker of, and I've put this in red, of cumulative cellular oxidative stress. This is the important part. You want to know how much oxidative stress is accumulating in the cell. So it is a chronic marker, not an acute marker. If it was an acute marker, it would be changing by the minute, and then it becomes very difficult to really know what is going on over time in the cell. So quantitatively, the 8-OHDG is assessing ongoing oxidative damage, and it monitors, in effect, the antioxidant therapies and detox that you may be doing and trying to do for the person that you're working with. And it's very simple collection. It can be done in dried urine. And so just remember that increased 8-HDG is associated with many different dysfunctions and diseases. It is looking at DNA oxidative damage, and it is in a cellular concentration. It is the established marker of oxidative stress, of aging, folks, aging, and carcinogenesis. And I put that at the end, very important. In other words, it will let you know if it becomes very high. It can be one of the early markers that there is a possible carcinogenic issue going on and that you could be getting into an issue of cancer. And what does it really do? Well, this is the double helix with the cross-linking. And what you need to know is that 8-OHDG is really working on one of the bases. There are 4 bases: guanine, cysteine, adenine, and thymine. And so it works on guanine. And what it is able to do is to check whether or not, because of stress, oxidative stress, you have basically disrupted one of these bases and its ability to function properly in the double helix. And what that does is it creates genotoxic instability, meaning instability within the genes, causes damage, which will do 2 things. Either it's so bad a damage that it causes cell death, otherwise known as apoptosis, or alters the cell's behavior, meaning some kind of possible malignant behavior, etc. And how do you deal with this? Well, just remember healthy lifestyles, balanced diet, smoking cessation. I don't even have to go through all of this. It's pretty obvious. Those lifestyles combined with various kinds of antioxidants, which many of you know very well, coenzyme, the various vitamins, melatonin, betaine, quercetin, etc. Curcumin, which has become much more popular lately, and of course, glutathione, which functions in the cell as an antioxidant. All of these will be able to solve the problem. So here is a case report. It's a 40-year-old male, and he complains of persistent chronic bloating and gas with constipation and occasional periods of just the opposite, of diarrhea. In his recent past, he has taken HCl supplements, betaine hydrochloride, to help alleviate persistent low stomach acids. Aha. Antibiotic treatment he's been getting for H. pylori infection. He says he's gotten partial relief. Remember the H. pylori. He was seen by nutritionists because he felt there needed to be some dietary assessment here. He's tried weaning off a high meat diet. Remember meat here, by consuming high amounts of legumes, maybe higher than normal, because he's been eating a lot of lentils, as well as large quantities of tomatoes, potatoes, etc. So you do a differential. What are the possibilities here? Well, the high protein intake, the gut dysbiosis manifesting itself in a number of ways, including H. pylori, metabolic dysfunction, and of course, always consider that he could have undiagnosed celiac disease or Crohn's. Many people are walking around with undiagnosed Crohn's. And of course, unfortunately, he could have colorectal cancer. So we would look at one of our reports. And why don't we just take a very quick look at reading a metabolic wellness profile? Well, the indican, these are all quintiles. Quintiles first, second, third, fourth, fifth, going from very, very low. And this is based on a population. Populations are in fifths. And so where does this person sit? He sits in a moderate level of indican, which is being shown up on the yellow box here. It's yellow because he's in the yellow area. And we also put moderate. Where is it in the reference range? It's in the moderate range. What is the actual numerical amount? It's 185 micrograms per milligram of creatinine. Why creatinine? Because you have to base it on creatinine because you're in urine. And we can go over that at a separate time. And so if you've got that level or even higher, here we're showing you one with very high levels of indican. What does it tell you? Inefficient protein digestion, very clear. There's very poor protein breakdown, and it's leading to amino acid deficiencies, poor glycemic control, hormone imbalances. Protein is so not well metabolized that it's putrefying. That leads to gut hypermotility, inefficient hydration. You need enough water in there, and it has to be able to stick around long enough with the food, but it's not, because the gut is sending that food out quite quickly while it's not being properly metabolized. That is one of the reasons you run into bloating and gas. There's indicators here for dysbiosis, SIBO, intestinal permeability, and of course, it can lead to liver congestion and poor detox. Some of the other testing that you'd want to do, MBA, the mucosal barrier assessment, which is one of the foundational FDN tests, food sensitivity tests for those who do them, the Candida test, we can go into that in some more details, and of course, testing hormones and SIBO. What about urinary total bile acids? Here's a second one. Well, here we're showing you one that's very high. We show you high, and we tell you how many nanomoles per milligram of creatinine. And here is one with very high. So what does it tell you? Well, there's liver dysfunction. The liver is congested. It's not properly pushing it through. There's very poor detox. So we don't know, is it part of phase 1 or phase 2? You need to determine that. There may be hepatocellular damage. Why? Due to drug use. A lot of people are using drugs, but they don't tell you how much, and they don't think it's a problem. Well, it is. They could be getting into a toxic overload, antigen and immune complex loads, etc. The detox protocols are well known. The water intake needs to be looked at. Exercise, sweating, you need to know how much water is leaving the body, how much you're taking in. Something which is becoming more and more discussed. Are they getting efficient sleep? Not just enough, but efficient sleep. Is there an anti-inflammatory diet? And that's part of the toxin reduction. And of course, the various testing, the MBA again, the mucosal barrier assessment, and the other tests which I mentioned earlier. And the last one here is, of course, the DNA oxidation, the 8-OHDG. And what are we looking at here? Well, here it's in the low level. Okay. Uh, what do we want to know? Well, here we're showing you one where it's actually high. Well, if it's high, what are you looking at? Well, immediately you say to yourself, well, there's cellular oxidative damage, and it's in the nucleus and in the mitochondria. Because remember, both the nucleus and the mitochondria contain DNA. The mitochondria has its own DNA. So it could be both of them being affected. With cellular oxidative damage, there's also a loss of lipid and protein function in the cell and therefore the body. If the nucleus isn't working well, how are the lipids and protein functions going to work well? They can't. And what you're going to get is accelerated aging. You now are very much at risk, if not already into diabetes, cardiovascular disease, ADHD. The list is very long. There are increased pathogens in the body, which the body can't get rid of properly. And so that creates immune dysregulation. And when those large guns are going off and the immune system is not properly balanced, that can cause massive problems. And so, what are some of the causes? Well, chronic dysbiosis. Don't forget, dysbiosis, especially when it is chronic, can cause major problems for the rest of the body. Heavy metals, other environmental triggers, all can be extremely important. What's the support? Well, one of the first supports is antioxidant support, because clearly there's an oxidation issue here. And so you've got to do that. Testing, MBA, the hormone analysis, you can do the stress and hormone profile, another one of the foundational tests of FDN. And then I'm not going to spend time on this, but then, of course, we go through how you can use both together. What if both are high? What if total bile acids and 8-OHDG are high? What if all, what about all three? Let's take this as an example. You've got high indican, you've got high bile acids, you've got sort of normal 8-OHDG. What's the clinical status? Well, this test tells you there's elevated liver load, clearly toxicity because the liver is not detoxing properly, and there's intestinal dysfunction. And those two elephants in the room need to be dealt with. So what's the action plan? Well, you want to support their digestion. You got to do whatever you can to at least try to make sure that they've got proper digestion. You've got to check that. You've got to support their liver function with supplementation, with many of the other things that one does for detoxing the issue of the liver. Investigate, remember, investigate their drug use, always. And I'm talking both medicinal drugs and recreational drugs. If there is a real problem, you may have to refer to various medical treatments and so on if they're getting to a very dangerous and level. And here's another one where they're all high. All of them are high. So elevated toxicity, free radicals, there's cellular damage, clearly increased load on the liver. You've got to deal with this. This is a fire that's there right now, and you've got to deal with that. So all of the things that I mentioned to you need to be done. You immediately need to initiate antioxidant protocols, definitely investigate their drug use, etc. And of course, all the other things which you could be looking at. One last thing I want to mention is if there's very low TBA, what if it's just extremely low? Well, there's very weak enterohepatic circulation. The areas of the intestine and the liver are not circulating properly. This could be due to genetic disorders. Maybe there's impaired bile production, and this person's had it for a long time. could be very well due to antibiotic use, because suppresses, antibiotic use suppresses the intestinal flora or changes it. And therefore, it reduces the levels of some of these deconjugating enzymes, which need a proper functioning microbiome. Biliary stasis, the bile is not functioning properly. They've got severe liver disease, cirrhosis. They're drinking too much, let alone anything else which is causing it. And that's going to cause low bile production. And then they're into a vicious cycle. And so what do you need to do? Well, the action plan is you got to look at all these things. Intestinal permeability, do the MBA test. You need to look at other pathogenic vectors. What else is going on? Food sensitivities? Yes, could very well be. Remember, as far as testing, mucosal barrier assessment, looking at things like zonulin, histamine, DAO, etc. And next month, we're adding LPS to that. Hormone analyses, and of course, comprehensive stool. So that was a very quick run through of this particular test. And there we are.
Dr. Gonshor, you did an amazing job as always. And it just reminds me of why we run that test on every single client. You're identifying three major components of health. You know,
Yep.
here we're not really looking to diagnose and treat a disease specifically, or just look at something. You know, you can say, "Oh, you have maldigestion. Let's take some enzymes and some HCl." That's too easy. With every single person, you want to get some data on digestion. Are you actually breaking down and absorbing food properly? Because you could be spending a lot of money on food. I eat all organic and I order my meat special online and things that I do. But if you're not getting it absorbed into the body, that's not good. And also, it tells you you have a dysbiosis. There's something going on with the immune system, your microbiota. So that test tells you so much. And on the digestion, detoxification, and then oxidative stress, all on one test, which is very reasonably priced. So it tells you so much about a person. Dr. Gonshor, on the bile acids, we used to measure that with lipid peroxides. It was more the breakdown of fats. Why is 8-OHDG the better marker for oxidative stress?
Well, remember that there used to be MDA. There are many tests that will look at that. I think what makes it most important, I alluded to earlier, and that is that it is a very reliable, reproducible chronic marker. In other words, unlike some of the other tests, which do move around a lot more and will change their levels, 8-OHDG doesn't. And so this doesn't make the others poor markers. I mean, MDA is a good marker. There's nothing wrong with it. But in the scheme of things, it's not as good. Now, if you were doing a plethora of liver biomarkers, which we're not doing here, then sure, you want to look at all three. You want to look at not just the oxidative issue in the nucleus, but you want to look at lipid peroxidation. You want to look at the protein function in the cell. But if you've got to choose, and here we're choosing, then you choose the one that is most reliable as a cumulative marker. And that allows you to know that in dried urine, remember, dried urine, let's remember this, dried urine, like dried blood spot, and even saliva, these are effectively spot tests. And so you're looking at something in a very small portion of your day. You peed, you put a little bit of blood on a piece of paper, or urine on a piece of paper, or you drooled into saliva for a few minutes. That very small element of time, you want to hope that what you're going to get in that time on biomarkers is going to be a reliable aspect of what's going on the rest of your day. And if it's not, then you shouldn't be using it. This is very different from doing cumulative tests, like we'll discuss some other time, whether 24-hour urine, if you want to look at hormones, etc. We can get into that later on. But here, in a spot test, a moment in time test, you want to know that what you're going to get on that paper when you start analyzing it is going to be a real window on what is actually going on with that person throughout their day.
I love that about the dried urine test, that you can get these three markers on it, which tell you an awful lot about a person. It also makes it really convenient. And of course, the price is good. It's great. And you could ship these kits all over the world pretty much and get the results into the lab or get the samples back to the lab quite easily. I love this test in combination with that blood spot, which for those of you listening, go back to the last episode that Dr. Gonshor and I did on the mucosal barrier assessment, because the two of these can be used together to give you a very complete picture, not only of digestion and detoxification, oxidative stress, but of the inflammation and even leaky gut. You've got your zonulin on that other test. So they're very effective. Well, I think that's it for my questions. We answered the others. I wanted to know about the main causes of protein malabsorption, but I think you went over it pretty well, unless you had something to add.
Well, I did want to stress something that you said, which is very important. When you talked about getting off red meats, for example, and saying, oh, I'm going from that to legumes, I'm going to add, lentils and etc. Remember that what I said earlier is that you've got to do everything in moderation. There's nothing particularly wrong with red meats. It's a problem if it's too much of your diet. In the same way that if you substitute that and go to lentils, and if you go to places like India, where I was last month, and their diets are filled with lentils. Well, wait a minute. If you're eating a lot of lentils, that's one of the things I showed you that can end up causing maldigestion, inefficient digestion, or the body just can't keep up in dealing with all of that. So the end product of putrefaction, etc., is the same. But you've switched one excess for another. So what's the message? Whatever you're going to do it in moderation. And that is extremely important. The only other thing I would add is very high protein diets. You've got to be careful, because not only are you going to experience pain, altered liver function, and so on, but you're going to end up with insufficient protein metabolism. The body can't break it all down properly. And if you add to that comorbidities, then you've got yourself a mess of problems that you need to deal with.
Fantastic. Well, thank you, Dr. Gonshor. For those of you who want to know more, all of this is covered very, very intimately and deeply in the Functional Diagnostic Nutrition Certification course, which you can find out more about by following the links around this video. And we'll look forward to seeing you next time, Dr. Gonshor. Can't wait till we talk about another lab pretty soon. Good to see you.
Good to see you, Lee.